All right, so now they'd figured out that DNA was the genetic material, they needed to figure out what was the structure of DNA, and that would help us figure out how DNA works. So in 1953, James Watson, who was an American biologist visiting Cambridge University, and Francis Crick, who was a graduate student, they introduced the double helical model for the structure of DNA. And there's a picture of them. Watson's on the right and Crick's on the left with one of their models of DNA that they built from whatever they could find, I guess. Now, of course, you can go and buy kits that enable you to construct DNA because the structure is well known. So let's have a look at their journey they took in order to figure out the structure of DNA. So Crick and Watson couldn't figure out the structure of DNA without the work from a biologist called Rosalind Franklin. Now, Maurice Wilkins and Rosalind Franklin, they were biologists working out of King's College in London, and they were using a technique called X-ray crystallography to study molecular structure. And they were using this technique to try and figure out the structure of DNA. Crick and Watson's approach was mostly to try and build models based on what they knew of DNA. So Franklin produced a picture of the DNA molecule using this technique of X-ray. And there's Rosalind Franklin on the left, and there's one of her X-ray crystallography diffraction patterns. And from this image, they were able to deduce a lot about the structure of DNA, and this image was critically important for Crick and Watson being able to finally figure out the structure of DNA. So they couldn't have done it without. Now, the X-ray crystallography showed that the DNA molecule was helical, and it also showed that the two DNA strands ran parallel, and Crick and Watson were able to deduce the width of those two parallel strands in the DNA helix, and they were able to figure out the spacing of the nitrogen bases. So from all of that information together, they were able to deduce that DNA is a double-stranded molecule forming a helix, and that's where we get this double helix from. So let's look more closely at DNA then. So DNA is a polymer, as you know. It's a nucleic acid, and it's made of 4 different nucleotides. Each nucleotide itself is made up of a sugar. In this case, it's deoxyribo sugar, and a phosphate group, and one of 4 different nitrogen bases. And the 4 nitrogen bases are adenine, cytosine, thymine, and guanine, and we abbreviate those to A, C, T, and G. Now, adenine and cytosine have a similar structure and are part of a similar chemical groups, and thymine and guanine have similar structures and they're part of a different group, as we'll see. Okay, so let's start then by drawing a nucleotide. All right, and I think it's important that you draw it. So draw it along with me. We've got a phosphate group, there's a P, and that's connected to a sugar, in this case deoxyribose sugar, which is a pentagon, and that sugar is connected to a nitrogen base. And there are 4 different nitrogen bases, remember. It could be an adenine, a thymine, a cytosine, and a guanine. So we can have 4 different nucleotides, and they only vary by what nitrogen base they have. So remember, a phosphate, a sugar, deoxyribose, and a nitrogen base, either adenine, thymine, cytosine, or guanine. All right. So just so I don't have to draw it every time, we've got a little graphic here. There's your phosphate group. There's your deoxyribose sugar, which is it's a pentose sugar. That's why we've got it as a pentagon. And we've got a nitrogen base, and they're all covalently bonded together. All right. Now remember that nitrogen base can be any one of the 4 different nitrogen bases. So here's a little bit more chemical detail to those. Here we've got our phosphate group, here we've got our deoxyribose sugar, and here we've got our nitrogen bases. In this case, we've drawn adenine, but it could be thymine, cytosine, or guanine. So have a little look at those for a moment, and you can see all of the elements in these different parts of the nucleotide. And just remember, at junctions like this and this and this and this, there are carbons, and here and here and here, and there are hydrogens also that are not drawn, to make it simple. So these nucleotides then are linked together, covalently bonded together, to form the two strands. So here we've got our 4 different nucleotides. There's the adenine nucleotide, and here's our guanine nucleotide. And if you remember, adenine and guanine are similar in structure. They've both got those 2 rings. All right, and there we've got thymine nucleotide, and there's thymine, and there's our cytosine nucleotide, and there's cytosine. Now thymine and cytosine also have quite a similar structure, and they're members of a different group. But these structural differences are important with the spacing of the helix. Alright, so the nucleotides are covalently bonded together, and they're linked between phosphate and sugar. So here we've got our nucleotide. If you remember, the circle here is the phosphate, the pentagon there is the sugar, and this up here is one of 4 nitrogen bases. So they're linked together, all right, and they're covalently bonded. So we go phosphate to sugar to phosphate to sugar. There's another nucleotide, covalently bonded, sugar to phosphate. All right. Okay, so here's our phosphate, here's our sugar, here's our nitrogen base in the nucleotide, and you can see that we've got in this what's known as the sugar phosphate backbone, alternating phosphate sugar, phosphate sugar, phosphate sugar, phosphate sugar. And that simply goes down one strand of the molecule, and then we've got our nitrogen bases, which form kind of rungs of a ladder, which we'll see in a moment. Now before I leave this diagram, I want to point out in the sugar these carbons. Carbon 1, carbon 2, carbon 3, carbon 4, and carbon 5 there. Alright, so they're important in letting us figure out which is the 3' end and which is the 5' end. So remember, carbon 1, carbon 2, carbon 3, carbon 4, carbon 5. Alright, and we'll come back to that later. So this would be one strand of the DNA molecule. We've got our sugar phosphate backbone, and then we've got our nitrogen bases sticking out to the right, which forms the rung. Alright, so this would be one strand of the DNA molecule, but DNA is a double-stranded molecule, so now we're going to construct our second strand. There's a nucleotide. Now notice that the second strand kind of runs in the opposite direction. So the nucleotide of the second strand is covalently bonded to another nucleotide, and then we've got one strand here, there's the sugar phosphate backbone, we've got another strand there, there's the sugar phosphate backbone, and these nitrogen bases are joined to each other by hydrogen bonds. The A's and the T's have 2 hydrogen bonds, and the C's and the G's have 3 hydrogen bonds. All right. Okay. And there you can see a little bit more chemical detail. Again, there's our sugar phosphate backbone. There's our sugar phosphate backbone. And notice how this strand on the left, everything, these pentagons are pointing up. And this strand on the right, the pentagons are pointing down. They run in opposite directions. And here we've got a hydrogen bond, or 2 of them between the A and T, and 3 hydrogen bonds between the C and G. Also notice that the thymine and the adenine are members of these 2 groups, and the guanine and the cytosine are members of two chemical groups. Now this, the fact that we've got an A and a T and a C and a G, this distance is the same. So that accounts for the two strands running parallel to each other. If you think about it, if a T was joined to a C, it would be a less distance here, and if a G was joined to an A, it would be a greater distance, and the strands might sort of run a little bit like this, as opposed to being completely parallel like that. Alright, so this obviously only shows 4 pairs of nucleotides joined together. All of your DNA molecules linked together would be 3 billion base pairs. Alright, so like a road, there's directionality along the two strands, as we've seen. One runs in one direction and one runs in the other direction. So we're going to have a little look at the directionality then. One end of the strand we call the 3' end, and the other end of the strand we call the 5' end. And each of the two strands has a 3' and a 5' end. The strands run anti-parallel to each other. That means in opposite directions but parallel. And this number 3 in the 3' end corresponds to number 3 carbon atom in the ring of the sugar. And the number 5 on the 5' end corresponds to the number 5 carbon atom in the sugar. So we'll have a look at that now. So I'm going to draw the sugar. The sugar kind of looks like this. We've got an oxygen, and there's a carbon here, and a carbon here, and a carbon here, and a carbon here, and it goes up there, and there's actually another carbon right there. 1, 2, 3, 4, 5. So this is carbon 1, carbon 2, carbon 3, carbon 4, and carbon 5. Now this carbon 5 is connected to a phosphate, and this carbon 3 is also connected to a phosphate. Carbon 1 is connected to a nitrogen base. Alright, so since this carbon 5 is up the top, this would be our 5' end, and since this carbon 3 is at the bottom, this would be our 3' end. And of course, if we were to flip these up the other way, and I'll just draw it small down here, there's our oxygen. Okay. Then we've got carbon 1, 2, 3, 4, 5. This would be connected to a phosphate. This would be connected to a phosphate. And this would be our 3' end. And this would be our 5' end. So hopefully that explains the ends. Now, understanding that there's a 3' and 5' end is very, very important when we look at DNA replication and when we look at how proteins are made from DNA. Okay, because I'm going to refer to 5' ends and 3' ends, because certain processes only run in certain directions. All right. So, DNA structure then is two anti-parallel strands that are twisted into a double helix. And there's a diagram showing the double helix. And these are the sugar phosphate backbones, which are not shown here. And there's the other sugar phosphate backbone. And when I called the nitrogen bases like rungs, it's kind of like a ladder. Sugar phosphate backbones. There's our nitrogen bases forming the rungs. And you can imagine you can take that ladder and you can twist it into this double helix there. And then pay a little bit of attention to these dimensions. Here we've got 3.4 nanometers. So this distance here from one bulge of the twist to the other is 3.4 nanometers. Tiny little distance. If you remember, a cell membrane is about 8 nanometers thick, and that T2 phage was well over 100 nanometers long. Alright, so here we've got a video showing the structure of DNA. What you need to do is just simply click on the video and you'll be able to see the video run and an animation of the structure of DNA, which is nice.